Welcome to this Edexcel GCSE biology revision session. Today, we'll cover past paper questions on the essential topics of cell biology, cell types, mitosis, meiosis, microscopes, and stem cells. Let's dive straight in with some questions and answers to help reinforce your learning. First up, let's review the basics of cell biology with some short answer questions. Here's your first question. What is the function of the cell membrane in animal and plant cells? The cell membrane controls the movement of substances in and out of the cell, maintaining the cell's internal environment. Great job if you got that one. Next question. Name 3 structures found in plant cells but not in animal cells. The correct answer is the cell wall, chloroplasts, and a large central vacuole. These structures help with support, photosynthesis, and storage. Now onto cell types. Here's your question. What is the main difference between prokaryotic and eukaryotic cells? Answer. Prokaryotic cells do not have a nucleus. Their genetic material is free in the cytoplasm. Eukaryotic cells, like plant and animal cells, have a nucleus containing their genetic material. Next question. Provide an example of a prokaryotic cell. Bacteria is an example of a prokaryotic cell. Prokaryotic cells are often single-celled organisms. Let's move on to mitosis, the process of cell division. Here's a question. What is the main purpose of mitosis? Mitosis allows for growth, repair, and asexual reproduction, resulting in 2 identical daughter cells. Another question. List the stages of mitosis in order. The stages are prophase, metaphase, anaphase, and telophase, followed by cytokinesis. Now let's explore meiosis, which is essential for sexual reproduction. Question. What is the end result of meiosis? Meiosis results in 4 genetically different haploid gametes, each with half the number of chromosomes compared to the parent cell. Question. How does meiosis contribute to genetic variation? Genetic variation arises from crossing over during prophase 1 and the random assortment of chromosomes, leading to unique combinations in gametes. Microscopes are vital tools in cell biology. Question. What is the main difference between a light microscope and an electron microscope? Light microscopes use light to view specimens and are limited in resolution, while electron microscopes use beams of electrons and offer much higher magnification and resolution. Question. Calculate the magnification of an image if the eyepiece lens is 10 times and the objective lens is 40 times. The total magnification is 400 times. 10 times 40 equals 400. Finally, let's discuss stem cells. Question. What are stem cells and why are they important? Stem cells are undifferentiated cells capable of becoming different cell types. They are important for growth, repair, and have potential in treating diseases. Question. What is the difference between embryonic stem cells and adult stem cells? Embryonic stem cells can become any cell type in the body, while adult stem cells are more limited in their differentiation potential. That wraps up our quick-fire revision on cell biology, cell types, mitosis, meiosis, microscopes, and stem cells. Let's look now in depth at cell biology and cell types. Question. Explain how the structure of a root hair cell is adapted to its function. Root hair cells have a long, thin extension called a root hair, which increases the surface area for water and mineral ion absorption from the soil. They possess a large vacuole that helps maintain osmotic balance, facilitating water uptake. Additionally, they have numerous mitochondria to provide the energy required for active transport of minerals into the cell. Question: compare and contrast the structures of animal and plant cells. Both animal and plant cells are eukaryotic, containing a nucleus, cytoplasm, mitochondria, and a cell membrane. However, plant cells have additional structures: a rigid cell wall made of cellulose for support, chloroplasts containing chlorophyll for photosynthesis, and a large central vacuole that maintains cell turgor and stores nutrients. These features are absent in animal cells. Next, we'll explore mitosis. Question: describe the role of mitosis in the growth of tissues, organs, and the whole organism in animals. Mitosis is crucial for producing genetically identical cells, enabling growth and development of tissues and organs. It allows for the replacement of damaged or dead cells, maintaining tissue integrity. In multicellular organisms, mitosis ensures that as the organism grows, cells divide to increase cell number, facilitating the development of complex structures and systems. Question: explain how uncontrolled cell division can lead to the formation of tumors. Uncontrolled cell division results from mutations in genes that regulate the cell cycle, leading to continuous proliferation without the normal checks and balances. This excessive cell growth forms masses called tumors. If these cells invade surrounding tissues and spread to other parts of the body, the tumor is considered malignant, leading to cancer. Now let's delve into meiosis. Question: explain how meiosis and sexual reproduction lead to variation in a population. Answer: meiosis introduces genetic variation through two main mechanisms: crossing over and independent assortment. During prophase 1, homologous chromosomes exchange genetic material, creating new allele combinations. In metaphase 1, the random orientation of chromosome pairs leads to different combinations of maternal and paternal chromosomes in gametes. Fertilization further increases variation by combining genetic material from two parents, resulting in offspring with unique genetic profiles. Question: describe the significance of haploid and diploid numbers in the life cycle of humans. Answer: in humans, somatic body cells are diploid, containing 46 chromosomes, 23 pairs. Gametes, sperm and egg cells are haploid, with 23 chromosomes produced via meiosis. During fertilization, two haploid gametes fuse to form a diploid zygote, restoring the diploid number. This alternation between haploid and diploid stages ensures genetic stability across generations while allowing for genetic diversity. Let's move on to microscopes. Question: discuss the advantages and limitations of using electron microscopes compared to light microscopes. Answer: electron microscopes offer higher magnification and resolution than light microscopes, allowing visualization of smaller structures, such as organelles and macromolecules. However, they require specimens to be placed in a vacuum and often involve complex preparation, which can introduce artifacts. Additionally, electron microscopes are expensive and not suitable for observing living specimens, unlike light microscopes, which can view live cells and processes in real time. Question: calculate the actual size of a cell if the image size is 5 mm and the magnification is 500 times. Answer: to find the actual size, divide the image size by the magnification. Actual size equals image size divided by magnification equals 5 mm divided by 500 equals 0.01 mm or 10 micrometers. Therefore, the actual size of the cell is 10 micrometers. Finally, let's look at questions that discuss stem cells in more depth. Question: evaluate the potential benefits and ethical considerations of using embryonic stem cells in medical research. Answer: embryonic stem cells can differentiate into any cell type, offering potential treatments for conditions like Parkinson's disease, spinal cord injuries, and diabetes. However, ethical concerns arise because harvesting these cells involves the destruction of embryos, raising questions about the moral status of embryonic life. Balancing the promise of medical advancements with respect for ethical considerations is a complex and ongoing debate in the scientific and public communities. Question: explain how stem cells are used in bone marrow transplants. Answer: bone marrow contains hematopoietic stem cells that can develop into various blood cells. In bone marrow transplants, these stem cells are harvested from a donor and transplanted into a patient with conditions like leukemia or lymphoma. The transplanted stem cells migrate to the patient's bone marrow, where they proliferate and differentiate to restore healthy blood cell production, effectively replacing the diseased or damaged marrow. So there you have 20 questions and answers from past biopapers to help you with your revision. Please like, share and subscribe to our channel for more videos to help you with exam revision. Thank you and best of luck.